Today is April 24th, 2017. My name is Sue Barman, and I'm at the Chicago McCormick Place, where we're holding the annual meeting of the American Physiological Society, Experimental Biology. Today I have the distinct pleasure of interviewing my PhD advisor, Dr. Robert D. Wurster, for the Society's Living History Project. Dr. Wurster is a long-standing member of the APS, and he's been associated with Loyola University's Medical School since 1961. Bob's research has focused on electrophysiological studies of neurons controlling the cardiovascular system, as well as the role of ion channels in brain tumor proliferation and studies in the spinal cord repair. And Bob, welcome to the Living History Project, and thank you for agreeing to this interview. I really look forward to it, and I hope you have looked forward to it too. Okay, is there anything you want to say in advance, or should we just go on? Okay, I'm going to ask you some questions. And so you were born in Mishawaka, Indiana.
Yes.
And you grew up in that state. And can you tell us about your family lifestyle and how you developed your interest in science?
Sure. My father was an eye, ear, nose, and throat physician and surgeon. Mm-hmm. And so experimenting associated with biological or medical systems was part of the lifestyle. He studied calcium influences on nasal sensitivity and allergies until shortly before World War II, which time he stopped doing his research. It's intriguing to me because he was studying calcium, and later on I was studying calcium effects on afferents or sensory fibers as well. My mother was a graduate of Butler University in biological science. She became a scientist for Eli Lilly, and this would be in the late teens. What she studied was insulin. About that time, Banting, Best, and McLeod were from Canada, had discovered that insulin played an important role in diabetes mellitus. And her task was for Eli Lilly is to determine what kind of dosages and how frequently would a person have to have the insulin to maintain a normal glucose level. So that was her task. She did that until her mother became seriously ill, and then she had to go home and take care of her mother on a full-time basis until she recovered. And fortunately, she did recover. But that took her out of the realm of the research lab. It's sort of interesting, she was invited back to Eli Lilly and to the labs where she worked at, and she saw on the ceiling that there was a purple stain or purple-brown stain, and she put that there, probably 30, 40 years before that. It was potassium permanganate, and it leaves initially it looks purple, but eventually turns to be a brown stain on it. So she felt very, very much at home to see that.
That's a great story.
She eventually, they moved to Mishawaka. They were both from the Indianapolis area, and they moved to Mishawaka after they were married. Part of their time before that, they were in New York City, where he did a residency in New York City, and then in Washington, D.C., and then moved to Mishawaka. So they were particularly my mother was a great advocate of science and a great inspiration for science. I latched onto that osmotically or some mechanism like that, and I became interested in science as well. Early on, I was interested in nuclear reactors, because at that time, nuclear reactors, this is post-World War II now, were a hot topic. And I had two cousins who were nuclear engineers, and they inspired me to follow in that direction. I didn't follow exactly that direction, but they certainly were an important influence on me. I went through school, but I was always interested in science when I was in grade school and in high school. And I would have projects, and I befriended the chemistry professor, I remember very, very fondly, and his encouragement of me to become, or to get involved in a career in science. So that was my sort of high school era. During that time, I was taking courses. I could take courses at Indiana University. They had an extension campus in South Bend, Indiana. That's right next to Mishawaka, where I lived. And I took courses there. And eventually those courses could be used for college accreditation. I also took courses at Notre Dame, which is near there as well. And they have excellent facilities and professors there. So that was sort of my educational background. When I got to my senior year in high school, I had only one course to take, and that was physics. And I started to attend the first couple of weeks of the physics course, and I decided that it was bluntly a waste of time because it was at such a low level, the physics, and I wanted to go on into things like engineering. My major in high school was engineering, pre-engineering program. So you took all the science and math and English courses that were required, but in addition you took machine shop and things of this sort, drafting and those kind of courses. So that was my major in high school. But I left high school at my senior year and decided I would go to Purdue. So I asked Purdue if they would let me come there, and they said yes, but you don't have a high school diploma, but we'll see how well you do. So that was very nice of them. And I started school at Purdue University and in the engineering program. I was in what is called engineering science. Engineering science are the theoretical aspects of all the major engineering areas like chemistry, mechanics, so forth. So nothing very practical about it. It was all theoretical, basically. But there were only 20 students for an engineering program at Purdue where there are many thousand of engineering students. 20 students, a program depended on 20 students. They eventually dropped the program, which case I had to choose another area and I chose biological sciences and chemistry. So that became my major at Purdue University. I don't know if you have any questions at this point.
Yeah, so I'm curious now, did you ever go back and get your high school diploma?
My mother insisted on that.
Okay.
And by that time, I was pretty aloof, go back to high school. I'm a college student, but I did, yes. I did go back almost 3/4 of a year later for the high school diploma, and my mother was pleased to see for that, so yes. But it's interesting, I remember very few of the students that were there. I was so involved, and I worked extra time with the chemistry professor who was very encouraging, so I wasn't, into very many other things, so even when I went to a couple class reunions, and no one knew I was there, and I didn't know anybody. I decided to give up on class reunions. Nothing against the institution. It's just.
So you had a very interesting background, obviously very supportive background of science, and even a high school that had all kinds of opportunities for
Yes.
you. And then going on to Purdue, and as you said, you then became interested in biology programs, and so you had opportunities to do research while in college.
Yes, I became a lab tech. When I'm not in class, that is mornings or afternoons or in weekends, for a physiologist. His name was Jake Wiebers, and he was an environmental physiologist. He studied the effects of environment on the human body, and I was his 60 cents an hour technician there or something like that. I think I got a raise to $1 an hour. But I didn't do it for money. I worked in the summers, so I actually ended up paying my own way through school. So it wasn't a money issue, it was an interest. I was interested in the science that he was working on. So he was very instrumental early on, Jake Wiebers was.
Okay, and then you ended up going to Loyola University to get your degree.
Yeah, that's sort of
And how did that happen?
Yes. Dr. Wiebers sent me to a symposium that was held in, I think, Columbus, Ohio. I don't know if it was at the university or not. And I got in the car and drove there. And I got to the outskirts of Columbus, Ohio, and I said, gee, do I want to sit through 3 boring days of meetings? I have a girlfriend in Cincinnati, I think, and that's what won out, and I made a right turn and headed south to Cincinnati. And that was a very important visit because eventually I married that gal and still married to her. So I gave up science, you see. But a very interesting thing, that meeting that I missed, a future mentor of mine, Walter Randall, was one of the premier lecturers for that. So I missed meeting him at that meeting. If I had gone to that meeting, I would have met him.
You would have actually met him earlier than you did.
I decided eventually that I was interested in a nerve action potential. And there was a biological science meeting there, American Institute of Biological Sciences. And they had their meetings, at least at those times, on campuses. Mm-hmm. And they had a demonstration of a sciatic nerve from a frog. And they stimulated, recorded, and I did my technical activities were early in the morning that day, and I saw them set it up, and it was working very nicely. They electrically stimulated this isolated nerve, and then there was a recorded electrical potential. And I came back after classes when I was to go back to the lab to work, and I stopped in to see, and that nerve action potential was still going on real strong. And that really made a big impression on me, and I decided I wanted to know what was the mechanisms that caused the nerve action potential. And I knew it was physical kind of things you had to study. So I went to a man who was a physicist. He got his degree at Purdue in physics. But he was studying in the biological science department there. And I went to him, I said, I'm interested in this nerve and those action potentials. And he said to me, young man, all that's okay, but the real action in the future is going to be DNA and RNA. And, I said, well, that's not really what I was thinking about doing. And this was not long after Watson and Crick's discovered in DNA. But I remember his recommending that. Well, he eventually went on and became a candidate for a Nobel Prize. He discovered the DNA sequence for the first living organism, which was tobacco mosaic virus. And so he was a very famous man. But by that time, he was at Caltech and no longer that way. But it was interesting because of his advice, and I guess I sort of ignored it at the time. I probably had cotton in my ears or something, figuratively at least.
Right.
But later on, he certainly was quite correct. Molecular biology is one of the main things in biological sciences today.
It certainly is.
So later on, I had to do a catch-up on the DNA and RNA.
But you still like that nerve action potential.
I still like that nerve action potential. So I then went to Chicago, great city that we're in right now, and I went to the University of Chicago, and I had contacts with the president of the dean of the medical school there, and he sent me to a physiologist there, and we talked about physiology, and he said, well, we don't do that kind of physiology there. You might go over to the University of Illinois, there in Walcott Street, over by the Cook County Hospital, and you might go in there and ask him. He said, while you're there, go down the street a block or two. It's a cruddy little building, but he said, there's an interesting guy there. And once you get done with Illinois, go down and visit with him. And that I did do.
That interesting guy?
Yes. When I visited the University of Illinois, it was the middle of the summer. People were on vacation, and I thought, gee, this is Dawesville. This is pretty boring, these people there. Hardly anybody around. Of course, they were all on vacation, but I didn't consider that. And then when I went down to Stritch School of Medicine, Loyola University of Chicago, there was Walter Randall, and he was surrounded. He sort of went with this cloud around him of enthusiastic students, both graduate and medical students. And he just radiated excitement about physiology. Eventually, he became like you, one of the presidents of the American Physiological Society. So he influenced me a considerable amount. And he was a great man, not only in the laboratory, but in his personal life as well. So that's always an inspiration to have somebody that is a real complete human, if you wish. I ended up not working directly with him. He was at this time going from temperature regulation studies. That's why he was at the Ohio State Symposium to talk about his temperature regulation work. But I was actually, but he had decided to become interested in the nerves to the heart. So he was just beginning, he had 1 or 2 publications. And he saw that, gee, you worked on temperature regulation and environmental things at Purdue University. I tell you what I want you to do. I want you to follow up on his grant and complete those, follow up on the research for those. So he turned that all over to me. He then pursued the cardiac and eventually got a program project grant for the study of innovation to the heart. And that made his career. And I, meanwhile, studied the temperature regulation responses. And so he did his experiments right in the office. And there was a man who became chairman at, wasn't Arizona, New Mexico, and chairman of the physiology department. And that was, Priola was his name, Don Priola. And we sat there, and then the experiments were just 3 feet away where they were working, so I began to get curious about what they were doing. But I did complete my doctoral work on environmental, the effects of temperature on the control of sweating. And I compared deep brain temperatures as much as you can record on a human, and we developed the tympanic membrane temperature system and the facial temperature system, which if we were wise, we would have patented because that's one of the ways people today use to measure temperature. Walt and I had a great time together when I went to Long Beach, California, and I went there to study reflexes, autonomic reflexes in spinal patients. And I went there, started the studies up. I lived in the VA, they gave me a room in the VA, and they had a collection of people who were volunteers that all had spinal cord injury, and I did a project with that. Walter Randall came there for 1 week and stayed with me, and we had a great time there. That was our closest time without any other interference, if you wish, together. So I really got to know the man quite well.
So this is while you were a graduate student?
Well, I was a graduate student, yes. And we published those works on the spinal patients. And it's strange because I recall those studies in the spinal patients, and I'm back now interested in spinal cord injury and how to repair spinal cord injury. So those experiences still
are part of your scientific career today.
Yes.
And so tell us a little bit about what it was like to be a graduate student back in that time, and how it might be different than it is now.
Yes, it is a bit different than it is now. We were in a cruddy old building. It used to be the Francis Willard Hospital. Francis Willard here in the Chicago area was the Women Christians Temperance Union, and she started a hospital in that building. It was eventually closed down because of it was unsanitary conditions. I don't know if you can fault Francis Willard for that or not. And next door to that place was the Morton Arboretum. There is a Morton Arboretum here. Mortuary School Morton School of Mortuary Science. In other words, where people learn to infuse bodies and undertakers. And I always thought that was funny because it was with the medical school there, the goal of the medical school was to keep people happy and living, and next door was the mortuary school. Also next door to us was the Chicago Medical School. And between the Stritch School of Medicine, that's where I was, it's part of Loyola University, still is today, and the Chicago, there was a concrete ledge that joined the two buildings. Obviously, at one time, those two buildings were attached to each other. And their dirt had accumulated up there. It was about 3 stories up, and trees started to grow up on that. And we called that the Gaza Strip, because they had mostly Jewish students at Chicago Medical School, and we had mostly Christian students. We had some Jewish students as well, but we called that the Gaza Strip that joined the two buildings there. Now, Chicago Med is moved up to the north suburbs of Chicago, and is the, I gotta think of her name now, but anyway, that's the, being with Walt Randall was amazing. He was always an inspiration. Physiology and the profession of physiology was a very important thing to him. So he made sure I got to the physiology meetings, like this meeting today. In probably 1963, 64, I had my first abstract at a meeting.
Was it an APS meeting? Was that
was an APS
the fall? Was it one of those fall meetings held on campuses at that time?
That's interesting. She brings up the fall meeting, or you bring up the fall meeting. That's something they don't do now, but it was actually sort of unique. The fall meetings were held on campuses. So you got to visit other colleges, and you stayed in the dormitory. And I could stay with my mentor, with Walt Randall, and I could share a room. But it was much more casual than it is. To come to a meeting like we are here in Chicago for the experimental biology meeting, they used to call that the FASIB, or the meetings. And that was much more harsh in a way, the discussions. And there were, one laboratory group was competing another laboratory group, and when somebody presented a lecture, they got up and argued, and it was very, very unfriendly, if you wish to say it that way. I don't think we do that in science anymore.
Right.
There's competition, certainly, but it's not competition that gets virtually nasty. So those meetings were different, but I liked the fall meetings. And the fall meetings, graduate students presented more, in addition to faculty there. So I thought that mix was much nicer.
Right, I had the same memories. I really enjoyed the fall meeting. Yes. My first meeting was the fall meeting of the
I think it became impractical when the university schedules got changed.
Correct.
And I think we could, before we had meetings in September, and universities started in late September, early October, and the dorms would not be free or available. So times have changed because of schedules like that.
Right. And so after you finished your PhD with Walt Randall, then you moved on to Germany for a postdoc.
Yes.
So what motivated the move to Germany? How did that happen, and how did that influence your career?
Those darn axons that I still had in my mind, and I wanted to record from very small afferents, or sensory fibers, and autonomic fibers as well. And those are what are called C fibers, and C fibers are about 1 micron in diameter. They're very difficult to record, at least in those days. And so I decided to look into what labs were actively doing that. Well, there are only a few labs in the world that were recording from C fibers, where you would dissect the nerve down to a single nerve fiber and record from a single nerve fiber. And that's a 1 micron fiber. There might be some other tissue around it, but it's still basically 1, 2, 3 or so microns. And that took a very precise dissection to be able to expose that, to keep it alive and to record from it. And I chose to go to a laboratory at that time that was in Salt Lake City, and as Ed Pearl was the laboratory, and Ed was one of the few people in the world. There was a group in Japan, a group in Germany, and there was a group in England, and Ed Pearl here in the United States who recorded from those. So I decided I'd like to become a graduate student there, and Ed Pearl said that I would be accepted there. But he was in the process of moving. He accepted the chairmanship at North Carolina, and so he was in the process of getting ready to move there, and I knew that I wouldn't see much of Ed Pearl. He'd be busy getting a Department of Physiology going at the University of North Carolina. So meantime, I discovered that there was a man in Germany who did these recordings from these small fibers. His name was Herbert Hensel. So I decided that I would go there and learn to do the recordings from these small fibers. And he was interested in temperature receptors. And they are small fibers. They are either C fibers or what's called A delta fibers, a little bit larger, but not very much larger. And he recorded from that. So I asked him if I could come and study those, and he said yes. I'd like to go back to California when I was with the spinal patients. When I worked with the spinal patients, I worked with them during the day. In the afternoons, what I did is I discovered that this was at Long Beach Veterans Administration Hospital, that at the VA hospital there, that there was actually a neuroscience institute had formed there. And I decided, gee, are there people that are interested in recording from nerves and things of that? And I met Doug Stewart, who was a scientist there. He had finished his PhD, and that was where his laboratory was there in the VA hospital. Eventually, the whole institute was moved to UCLA, and he's there today as a institute at UCLA. But interesting, I went to see Doug Stewart, and I don't know if you've ever met Doug Stewart
Yes,
or not.
I have.
But it's the kind of person you don't forget.
Right.
Australian, an Olympic athlete. He was a high jumper for Australia, and he had a great Australian accent. I think he's still around. I hope he is. But he was a great inspiration. And I would go there in the evenings, and they were just getting their experiments going, and they were recording from the spinal cord. And I got interested in the recordings from those nerves. So remembering those experiences, and Doug Stewart had me he moved to Davis, California, and then to University of Arizona, where he was chairman of physiology at Arizona. Doug Stewart always invited me to come there and visit with him. And so he was always an inspiration to me. So I remember those things. And when I went on to think about going to Germany, I said, gee, it'd be nice if I could do some recordings, intracellular recordings from neurons in the spinal cord and take a look at that. And I discovered there was a laboratory in Germany that was developing these techniques, and it was in a town called Bad Nauheim, and it was a Kirchhoff Institute. It's part of the Max Planck Gesellschaft auf Deutsch, but it's a super institute, and they have 30, 40 different institutes scattered around Germany today. But these are research institutes, and so I asked them if I could also come and work in their institute there, and they said yes. And I wrote an application to NIH, and NIH funded the application for a fellowship. I was told that it was the only fellowship that, I don't know about NIH, but at least the National Heart Institute, as it was called in those days, the National Heart Institute gave one fellowship for someone outside of the country. So I felt very honored. And I went there first in Marburg, where I worked with Herbert Hensel, and then to Bad Nauheim, where I worked there in the laboratory. Interesting thing, I was so concerned about Ed Pearl not being there when I arrived. Well, it ended up the man I wanted to work for or work with in Germany, he had been the head of the university, the rector of the university for 2 years, and he stepped down. They had 2-year limitations on how long you could be rectors in those days of the university. And the next person to become rector was a person who couldn't handle problems. So anytime problems occurred with the students, and in those days, this is 1967, there were riots in the streets, and the new rector of the university, not my mentor, managed to disappear when the students were rioting. And Herbert Hensel had no problem getting along with the students. He'd go out in the middle of the riot and calm the students down. and let's have a meeting, let's resolve this in another manner. So he was preoccupied with that, even though he was no longer the rector. So the first half of my period there in Marburg, I actually didn't work with him, so I had to learn to develop the technique, which I did. He then returned to the lab, and he approved of my technique. So it was probably a good experience that he wasn't there, because I could learn.
Develop your independence.
That's correct.
And
So he was quite an inspiration to me. In Bad Nauheim, I then went to Bad Nauheim after my stay in Marburg and worked on spinal cord. And we recorded with microelectrodes from motor neurons. And actually, we recorded from a cell that's called the Renshaw cell. And the Renshaw cell controls the reflex sensitivity of the muscle. So when you tap the tendon or the muscle and you get a reflex, it regulates the motor neuron excitability. And it was the first time they had been intracellularly recorded from. So that was interesting that I had that, that opportunity to work with it. And that was in the laboratory of Peter Pirau. It was Friedrich Karl was his technical name, although we called him Peter. And Peter became a friend until his passing, 10 years ago, something of this sort. And Peter came to my laboratory in Chicago.
While I was there.
While you were there. I went back to Bad Nauheim to work with him.
During my last year as a graduate student. Yeah. And so, but transitioning from the postdoc to your faculty position, so yes. You had opportunities to stay in Germany or come back to the Chicago area, and Walter Randall invited you to come to be a faculty member.
Well, sort of. He invited me to come back. I wanted to come back to the United States. After all, my fellowship was funded by the American people, really.
Correct.
And I think I have a responsibility to bring technology and whatever I learned back to the United States. Peter was very helpful in me getting that. But Walt Randall said, well, why don't you come back to Chicago? We can use some help here. And meanwhile, you can go and look around at universities and to see about where you want to go. So I was a temporary employee. And I tell people that I've been a temporary employee of Loyola University for 50 plus years, whatever it is. He then invited me to stay there, and I helped him get a program project. That was his first program project grant, a big grant. It was like $5 million. In those days, that was a horrendous amount of money for a whole group of people to do research. And I had a section of that I took care of.
And that funded my PhD work and my stipend.
That's right. Thanks to Walt Randall.
The stipend all of $265 a month that went up to $325 the last month of my.
We overpaid you, but
Yeah, today that's a
No, I didn't. I take that back.
So tell us a little bit about how your career transitioned when you became a faculty member at Loyola, and you really embodied a lot of different areas of neuroscience. Obviously, neuroscience is the center of your research, but
right.
It wasn't just a simple. You obviously started temperature regulation. You've done spinal cord injury, a lot of things. So tell us a little bit about all the opportunities that you had, the interactions that you had with various people.
That frog axon was still.
That's still in your mind, yes.
See, what a little, 30-minute observation of something like that persists still today,
Yeah.
in fact. So I decided I wanted to focus on neural control and neural responses. Initially, neural control of the heart. And you were an important part of those studies, along with Bob Foreman and a number of other people there. So we had great times there. I became interested in other aspects of the nervous system. I eventually befriended the new chairman of neurosurgery, and he invited me to become a faculty there as well. So I had dual appointments in neurosurgery and in the physiology department. And I started a neuroscience graduate program there at Loyola University, which is still running today, and helped start the Neuroscience Institute there at Loyola University. So more focused on things outside the department, nothing wrong with the department, but my interest got distracted, if you want to call it that, or diverted to, is a better term, to neurological surgery. In the Department of Neurological Surgery, I got involved in residency training and eventually became the director of the residency program there. That's a little peculiar, and it even gets more peculiar in just a minute, for a basic scientist as myself in a neurological surgery department. But I worked with the residents. We taught them about applying their knowledge to the clinical situation. I didn't tell them what to do on clerkships, obviously not. But I became the director of that. And eventually I actually became not the chairman, but the vice chairman of the department, which is really peculiar. And I'm sure some of the neurosurgeons in the department say, what's going on here? But the chairman of neurosurgery, Howard Reichman, got along real well. And so we had this connection for many years. Until I retired, I was dual appointments there. I also got involved in projects from our engineering group at the Hines Veterans Administration Hospital, which is right next to Loyola's new campus, they eventually moved out of Chicago to Maywood, Illinois. And I got involved with an engineering group, Rehab Research and Development Center, and was offered the directorship of that. I didn't follow up on that. I wanted to I thought it would distract from my laboratory activities. But I still have a connection with those projects there, and continuing. Friday I'm going to meet and talk more about a couple projects there at the Hines VA. I was about a third of my time at Hines VA, and the other 2/3 were at Loyola.
And one of the other things I remember is your lab was one of the first ones to use a computer for data analysis while I was a graduate student. And talk about that computer. Yeah.
When I did the work on spinal patients, and the temperature reflexes and things of this order, we were collecting huge realms of data, and we said, well, we can't do this. Remember, this is 1963. And I said, there's these things called computers. And so with the help of a friend of mine, Bob McCook, we converted the laboratory to a computerized laboratory. So all our data came out not as photographs or on ink charts, but on punch tape, computer punch tape. So those days I didn't have a computer. I took it downtown Chicago. Statistical Tabulating was a company that had a computer there, and I processed my data there. I had to write the programs on it. And eventually we decided, well, we should have a computer of our own. The university had one computer. And we decided that we wanted to buy a computer for our own. So Dr. McCook and I went off to Digital Equipment Company, which is in Maynard, Massachusetts, and we took their computer training course there, learned to program their computers. Of course, those computers are much simpler than the computers are today, but then we brought back a digital computer, which was a huge monster.
Took an entire room.
Took an entire room, had to have air conditioning for it. It was a transistor, they had transistors that were finite elements that were transistors, not little chips inside those. And we learned to program that and to teach programming languages there on the Digital Equipment's computers. But there reached a time that I had to say to myself, Bob, you're spending so much time on computers, maybe you should just change your career and work on computers, or get someone else to work on computers and you continue to focus on physiology. I chose the latter, and I said that someone else can follow that technology. That's never going to lead to anything anyway for a career. Little did I know, of course. If I had followed that pathway, who knows, where I would have been.
And so, what do you think has been your greatest scientific achievement?
My greatest scientific achievements were training graduate students. That's the greatest, and I'll add postdocs as well.
Right.
I think that's the greatest achievement. What you do in science may seem big today, but tomorrow something else is going to come along. So it diminishes in its size. This is figurative size. It diminishes with time. Sometimes you do things that someone later on, 5 years later, proves you're wrong or whatever. That's part of the scientific process. But when you train graduate students, those are, and think about those go on to train others and others, it's a family line that you're starting. It's like having a family and initiating, and here's the newborn child, the graduate student, not a child by any, but you get the idea. I think that's the greatest achievement.
Yeah, I agree, and I think when you talk about a family, I mean, I know definitely for me, my experience it was at Loyola,
Yes.
working with you in Randall's department.
Right.
It was a family. I mean, it really was. We all felt that way. It was a small enough program that everybody was comfortable together. And also, as you mentioned, when you started graduate school with Walter Randall, he had you become a member of he had you go to physiology meetings, APS meetings. And I also recall that he had all of us become student members of APS when we passed our comprehensive exam.
Yes.
And so what has it meant for you to be a member of the APS for so many years? What is the society, how has that helped your career?
Well, it was a basis I could always come back on. It was connections to fellow scientists. It had an important part as a graduate student too, though, as we were talking about going to colleges and participating in conferences that the American Physiological Society was backing or promoting. So I think it's good to have a professional association, an organization which represents you. They're representing, when you get a grant, you assume that, well, that's because you wrote such a great grant or you're doing such good research that the federal government is willing to put money. But behind that grant is a lot of the American Physiological Society is very important in maintaining the respect of science to keep the funding going. We see that today. It's a real challenge today in today's world. The reality is that science is influenced by political things. We need representation. We can't all spend our time doing that. And the American Physiological Society has been that, still today.
Right. And what kind of advice would you have to young trainees today as they're entering the field of physiology?
Focus. Focus. Keep your looking for other new ideas with it, but I think you really have to focus. You may not want to spend your whole time studying this small little object here, but when you're doing that, you have to really say, this is everything. I put aside everything to look at this, to do these experiments, to write this paper. I think its focus is the most important thing. I think to share with others ideas and other faculty. I can recall other faculty at Loyola that were very important for stimulating ideas. And they themselves are probably not, famous scientists or something of this sort, but they play an extremely important role. But you have to have the focus on it.
Very good advice.
Yeah.
Now, you obviously spent a lot of time thinking science. Your life, you've described it as, and you still do after all these years. And you've obviously been very committed. You went through school in, 3 years of high school, 3 years of being in college and that kind of stuff. You've gone very fast. But outside activities, you've done things that are interesting outside as well, right? You must enjoy things other than science.
Yeah, I like Lake Michigan out here. It's a great place for sailing, and I love to sail. I taught sailing to individuals.
You tried to teach me how to sail.
Yes.
Yeah, it was a disaster.
A few years ago, I taught a graduate student how to sail. He came to, we have a little home in Michigan on a little lake, and I took him out in the boat and showed him how to sail. And he was out there sailing for 5-6 hours, a long time. That was his first time sailing. The next week, this individual rented a sailboat, a big sailboat, 30-foot boat. I don't know, somebody's out of their mind to rent a sailboat to someone to take out in Lake Michigan. And he sailed it all the way across to St. Joe Benton Harbor and sailed it all the way back. After 3 hours on a little dinghy. No, I like sailing. I also like art. When I was a boy, I attended art school in South Bend, Indiana. They had a very good art school there. And I attended art school and learned a lot about fundamental things, about perspective, lighting, and things of this sort of, that are important for art. But I reached a point where I had to decide, gee, are you going to be able to earn your living as an artist? That's a rough life to get started, to get a reputation out, and the percent that are successful at that is probably under 5% or something of this order. And so I figured that I'd probably starve as an artist, and that I better stick, I had a forte for math and science, I better stick with science, and that's a better aspect. But today I'm back into art now. And so I spend time painting and wood carving. More recently, more wood carving than painting. And we have one of our past presidents here, George Bush, who is a painter, and he's a pretty good painter. Surprisingly, I like him as a painter better than as a president. But some people say maybe your painting's better than your science is. Well,
you've made great contributions to science, and
thank you.
This has been a great opportunity to learn some things that I didn't know about you, Bob. It's really been fascinating. I hope you've enjoyed it as well.
Thank you.
So thank you very much.